Welcome everybody to the living component of our study. You're almost done having to look at me behind this podium. I'm going to talk now. Raise this up, thank you. I'm going to talk now about changes in Periphyton or lack of changes in Periphyton across our treatments. First, a little bit of a definition. We're talking about Periphyton. We're talking about any freshwater organisms that are attached to or clinging to the plants or the bottom substrates projecting above the bottom of the stream channel, and that could be living or dead organisms. Periphyton represents the base of the food chain, and given the association of Periphyton to the bottom sediments or the benthos, they are affected by chemical and physical processes. They also have a short life cycle and a rapid reproduction rate, so those characteristics together make Periphyton potentially a valuable indicator of short-term change to the environment. Production in headwater streams is primarily heterotrophic in that it relies for the most part on external inputs from the canopy. Therefore, autotrophic or primary productivity is very low in the headwater streams compared to the larger downstream systems. Previous research in this area has identified, for the most part, that light is the limiting factor in the growth of Periphyton. Some research has also identified an increase in streams that were bordered by a clear cut or a very narrow buffer. And other factors that have been found to limit Periphyton: temperature, sediment flow, nutrients, and consumers. Our objective here to evaluate the response of Periphyton total biomass as well as the algal component of Periphyton across our varying buffer treatments. And once again, was there a harvest response? Was there an increase, decrease in all of the harvests, in all the treatments compared to the reference? And then was there a between treatment response? Our metrics for assessing Periphyton and treatment, looking for treatment effects on Periphyton, were total biomass, which was measured with an ash-free dry weight, and the algal component of the Periphyton community, which was measured with a chlorophyll A. We also, after the fact, a post hoc attempt to try to explain some of the results we were seeing, added a few more metrics. Namely, we took a look at some of the data values that we got for shade versus both ash-free dry weight and chlorophyll A, and Periphyton versus sediment on our tiles. I'll talk about that a bit more in a minute. But we plotted some of those variables against each other and looked for correlations simply by adding a trend line and seeing what the R-squared values were there. For this work, we did have 3 pre-harvest sample years along with the 2 post-harvest years. We used artificial substrates, namely unglazed ceramic tiles that we anchored to the stream bed with some rebar, put some spacers in there to try to get them up and off of the bottom of the stream bed as best we could. We installed 4 pairs of tiles at each site. All of them were in the main stem channel. And so 2 of those pairs were in areas that would have been buffered had it been a forest practices treatment. And then the other 2 pairs were in areas that would have been buffered if it had been a forest practices treatment. All of our stations were at least 20 meters away from a change in buffer type and at least 20 meters away from any roads. We had 2 sample periods each year, early summer and late summer. It came time to collect and process the tiles. The first thing we did out at the site was note if any part or all of the tile wasn't dry. In that case, we excluded that sample from any further analysis. We also did a visual estimate and recorded the percentage of each tile that was covered in sediment And for the most part, we're talking about organic flocculent material over our tiles. See a little bit of that in that picture. We carefully removed each tile from the site, tipped them to remove the non-periphyton material, the debris that was on the tile, put it in a Ziploc bag and made sure to keep it cold and process them in the lab during the same day. As far as the actual processing, essentially we used wire brush to scrape off each tile into a wash basin, and then scraped it clean with a razor blade, rinsed it a few times in between each step, and then rinsed out that basin solution into Nalgene bottles, which were again put back into the refrigerator until they were picked up for processing the next day. At that time, we also measured the tile surface area, and we randomly assigned one of the two analysis treatments, starting with the very first tile and then alternating from there. And again, our two sample processing techniques were ash-free dry weight, which involved filtering the sample from the solution, drying it out to a constant temperature and igniting it at a really hot temperature, re-wetting, re-drying, and then calculating that weight at the end. And this work was contracted through the Manchester Environmental Lab. Chlorophyll A also involved filtering the solution, preserving it, extracting pigments, and then determining the fluorescence with a fluorometer. And the proportion of algae that you're sampling with chlorophyll A is relatively constant. And so in that regards, that particular measurement serves as an index of algae. We standardized our sample results as total volatile material, which was simply the result times the sample weight and divided by 1,000 to get that on a grams per liter scale. We adjusted each sample by the tile area so that we could see the total volatile material per area. And then we averaged those results for each site and sample period in year. Same null hypothesis. Same mixed effects model, our focus again on the period by treatment effect. And then our post hoc evaluations of periphyton versus shade and periphyton versus sediment. So since this was an attempt after the fact to try to explain some of the results we were seeing, we didn't have direct measurements of shade over our tiles. And so we pulled that information from the nearest Department of Ecology shade station. We looked at both densiometer values at stream level as well as effective shade, but here I'm just going to show you some of those comparisons for the densiometer values. And then we plotted the sediment value that we recorded in the field, covering each tile versus the periphyton result on that tile, and calculated some R squared values of those trend lines. So our results. The short answer is we did not see any significant treatment effects for either ash-free dry mass or ash-free dry weight, chlorophyll A, or sample period. But for ash-free dry weight, we did see an increase all across the board for both sample periods. Chlorophyll A, in the early summer, it did appear as though there was a little bit of an increase in the reference compared to the other sites. But again, that did not prove to be statistically significant, and even less so during the late summer period. So here's where we plotted the values up from those densiometer readings at shade level in comparison to the ash-free dry weight. A couple things that you'll see from this graph. First, a very bimodal distribution for our shade values. We had quite a few samples that were a little under 40% total shade as estimated by the nearest densiometer station, and then a lot of values up above 60 or 65%, but really very few shade values in between, regardless of whether those samples were in a reference reach, buffered reach, or unbuffered reach. I should also mention that we only pulled the nearest shade information from a similar buffer configuration. So if a tile was in an unbuffered reach, we only use the nearest shade information from an unbuffered reach. R-squared values were tiny. These plots are really not explaining any of the variation in our data. Same story with chlorophyll A, again a bimodal distribution in our shade values and not much in the middle there. Again, at most 13% of the variance in our data was explained by plotting shade versus chlorophyll A. Sediment graph is essentially the same. I've just color-coded it a little bit differently by treatment instead of by buffer type. We had no way of knowing when we did record the sediment cover over our tiles how long that sediment had been there. And so there's nothing we could really do about it. But we wanted to take a look and see if we saw any correlations between sediment and biomass, and if as a result of that we should try to adjust our sample area, for example, by the portion of it that was covered in sediment. We did not see any correlations in that regard, but we did note that nearly 78, a little over 3/4 of all of our samples did have some sediment on them. So there's probably more to the story here, and perhaps some other ways that this information could be teased out in the future. And so to summarize, there was no evidence of a treatment effect on either total biomass or the algal component of Periphyton. No, we did not find any correlations between shade and Periphyton. Little seasonal difference in Periphyton, and no correlation between the total volatile material and sediment cover for either of our Periphyton analysis methods.